I have the pleasure of introducing Nebeyat Zekaryas, who's entering her final semester at Diablo Valley College. After she gets her associates, she will pursue a career in nursing. The title of her project is Investigating Mineral Micronutrients in Lungs of a Sickle Cell Mouse Model. A fun fact about Nebby is that she has lived in 5 states.
So today I'm going to be presenting you what I've been working on this summer with Hart Horniman and Dr. Angela Rivers, which is investigating mineral micronutrients in the lungs of the sickle cell mouse model. So sickle cell disease is caused by the inheritance of mutated hemoglobin. The inheritance of one altered gene is known as sickle cell trait. And individuals with sickle cell disease suffer from chronic hemolytic anemia, painful crises, and shortened lifespan, as well as multi-system organ damage. The known pathophysiology of multi-system organ damage is the deoxygenation of red blood cells, which creates the sickling shape, which does lead to restricted blood flow. In sickle cell disease, these are the following lung complications associated with it: acute chest syndrome, asthma, and pulmonary hypertension. Acute chest syndrome refers to conditions of the lungs that are identified by symptoms such as hypoxia, cough, fever, chest pain, and lung infiltrates that ultimately cause a vaso-occlusion in the blood vessels, and is the leading cause of death in people with sickle cell disease. Many patients with sickle cell disease suffer from asthma, and there's evidence to suggest that it is more pervasive in individuals with sickle cell disease than in the general population. Pulmonary hypertension is another common respiratory disease seen in sickle cell disease. And it occurs when blood pressure between the heart and lungs is too high, and the high pressure results in narrowing of arteries, which can in turn lower the oxygen levels in the blood. Okay. The most common mineral micronutrients that have been studied in sickle cell disease are copper and zinc. Patients actively experiencing vaso-occlusive crises were found to have higher levels of copper in their bodies than control patients or trait patients. Zinc deficiency is common amongst children with sickle cell disease and has been found to be associated with increase in pain crises. Zinc serum levels have been found to be lower in subjects when actively experiencing a pain crisis at the time of data collection. And elevated copper to zinc ratio in sickle cell disease patients is associated with increase in sickle cell oxidative stress. We hypothesized that there would be different proportions of micronutrients present in the lungs of mice with sickle cell disease, sickle cell trait, and control. And our specific aims were to determine the proportions of micronutrients in the lung of sickle cell, trait, and control mice. These are the methods we used to accomplish our specific aims. Lung samples were collected from control, trait, and sickle cell mice. The samples were weighed and then dried in an oven for 24 hours. The then dried samples were weighed again and then dissolved and digested for 16 to 18 hours. These digested samples were then diluted and the remaining particulates were removed by centrifugation, and the samples were then analyzed by the ICP-OES. And the concentration of micronutrients in each sample was then determined using a standard curve. Our results found that the zinc showed no significance. However, copper was greater in the sickle cell than both the trait and the control, with a p-value of 0.0054. And the copper zinc ratio showed significance and was greater in the sickle cell mice with both the sickle cell mice and the trait mice with a p-value of 0.001. In conclusion, the sickle cell disease mouse model can be used to study micronutrients in the lung. Copper levels trended higher in the sickle cell disease mouse model lung, and we hope to use these studies to point to a new mechanism of pathophysiology in the lungs. In terms of acknowledgements, I'd like to thank Dr. Angela Rivers and Hart Horniman for all their support this summer and their patience throughout this process. I could not have done this without them, and then as well as this research being funded by NIH.
Fantastic, really interesting. One of our first animal model studies that is presented today. Questions that we have for Nebby. Lots of, there we go. Barry, you got a question.
I was wondering why copper and zinc cause an increase in the crises. Also, kind of like future steps like the pathogenesis to yeah.
So the copper causes an increase in the reactive oxygen species, which increase in the ROS leads to molecular damage, which in sickle cell disease is oxidative stress, is expressed as oxidative stress.
Nebby, you alluded to the fact that this is a multi-organ damage occurs in sickle cell, and we've heard about that a lot this summer. Curious if the mouse can be used to look at different tissues and if that has been done in the lab.
So this is because of the prevalence of lung complications with sickle cell disease patients. That's why we chose to focus on the lungs. But we are now actually collecting other organs and, trying to see the levels of different minerals in those as well.
Excellent, interesting. Catherine?
Yeah, so kind of going off of Barry's question. So in that slide, you were talking about how copper and zinc are both associated with certain symptoms of sickle cell. However, in your results, zinc was not, the difference in zinc between the three was not found to be significant, but the copper zinc ratio was found to be significant. So kind of is that, what do you make of that? I'm kind of unsure as well.
So my understanding is that copper zinc ratio is used as like a predictive marker of oxidative stress, but the copper level, the ratio itself, the copper level has to be higher than the zinc. This has been studied in the blood of humans, but this is kind of like preliminary looking at what the levels are like in the organs themselves.